Welcome to Frost and Sullivan's Growth Podcast. In this episode, we examine how RNA therapeutics are creating new possibilities for treating rare diseases at their underlying genetic source. More than 7,000 rare diseases have been identified, yet fewer than 5% have regulatory-approved treatments. Around 80% of rare diseases have a genetic origin, making RNA-based approaches particularly relevant for patient populations that have historically had limited therapeutic options. Antisense oligonucleotides, small interfering RNA, and messenger RNA are already demonstrating different ways to modify, silence, or replace disease-related biological processes. The scientific potential is significant. The challenge now is translating that potential into targeted, scalable, and accessible treatments. So, where is RNA innovation heading next?
Rare disease treatment is moving closer to the genetic cause.
For many rare diseases, treatment has traditionally focused on managing symptoms. RNA therapeutics offer a different possibility. Because many rare diseases originate from specific genetic abnormalities, RNA therapies can intervene closer to the underlying biological mechanism. Antisense oligonucleotides, or ASOs, can alter RNA processing or reduce production of disease-associated proteins. Small interfering RNA, or siRNA, uses RNA interference to silence specific genes. Messenger RNA, or mRNA, takes another approach by providing cells with instructions to produce therapeutic proteins. These modalities are already moving beyond experimental science. Approvals of therapies such as nusinersen, patisiran, givosiran, and tofersen have demonstrated the clinical potential of RNA-based approaches across different rare diseases. That validation is strengthening confidence in RNA as a therapeutic platform and expanding the question from whether RNA can work to where it can create the greatest clinical value.
Different RNA modalities are finding different therapeutic roles.
There is unlikely to be one RNA modality that addresses the entire rare disease landscape. Each platform brings different mechanisms, delivery requirements, and disease opportunities. siRNA development spans hematologic, neuromuscular, metabolic, liver, immunologic, and central nervous system disorders. Tissue-targeted delivery and well-defined molecular pathways are helping expand RNA interference beyond established liver applications. ASOs have particular relevance across neurodegenerative, neuromuscular, ophthalmic, metabolic, respiratory, and ultra-rare genetic disorders. Their flexible sequence-specific design also makes them particularly interesting for personalized therapies. mRNA approaches are opening opportunities across rare respiratory and metabolic disorders, where protein restoration and lipid nanoparticle-enabled targeting could address limitations associated with conventional protein replacement. For developers, the strategic challenge is therefore selecting the right modality for the disease mechanism, target tissue, and required therapeutic effect.
Personalized RNA therapies could redefine drug development.
One of the most interesting developments is happening at the smallest patient scale. The modular nature of ASOs is enabling N of 1 and N of few therapies designed around mutations affecting individual patients or extremely small populations. That represents a very different development model from conventional drug development. Advances in genome sequencing are making it easier to identify disease-causing mutations, while evolving regulatory pathways could support more individualized therapeutic approaches. But personalization introduces its own challenges. Developers still need to address evidence generation, manufacturing scalability, patient identification, and diagnostic delays. The opportunity is significant, particularly for ultra-rare diseases that may never support conventional large-scale drug development. The question becomes how the industry can build development models that make highly personalized therapeutics scientifically rigorous and commercially viable.
Delivery remains one of RNA's hardest problems.
Scientific progress alone will not determine how far RNA therapeutics can expand. Delivery remains one of the most persistent barriers. This is particularly important for rare neurological diseases, where therapies need to reach the central nervous system and overcome the blood-brain barrier. Emerging approaches include BBB crossing delivery technologies, lipid siRNA conjugates, brain shuttle systems, and engineered nanoparticles. The objective is to improve tissue specificity, stability, durability, and ultimately the ability to deliver RNA therapeutics less invasively. Progress here could materially expand the addressable rare disease landscape. For developers, delivery technology may therefore become as strategically important as the RNA modality itself.
AI could change the economics and speed of RNA innovation.
The other major opportunity comes from artificial intelligence. AI and machine learning can support several stages of RNA therapeutic development, from target discovery and sequence design to delivery optimization and toxicity prediction. That is particularly relevant in rare diseases. Small patient populations create pressure to identify targets, optimize candidates, stratify patients, and design trials more efficiently. AI could help integrate multimodal and clinical data sets, improve rare disease target identification, and support more predictive approaches to safety and therapeutic design. Combine that with advances in tissue-specific delivery, and a much larger opportunity begins to emerge. The convergence of AI-enabled drug development and targeted delivery could allow companies to develop RNA therapies faster while reaching disease areas that remain difficult to address today. The organizations best positioned for this next phase will be those that can align the right RNA modality with the right patient population, improve development efficiency, and overcome the delivery barriers that still limit clinical reach. Thanks for joining us on Frost and Sullivan's Growth Podcast. RNA therapeutics are opening new pathways for genetically driven rare diseases, with progress across ASO, siRNA, and mRNA platforms expanding the possibilities for disease-modifying and personalized treatment. The next phase will depend on advances in precision diagnostics, AI-enabled development, and tissue-specific delivery that can translate scientific progress into viable therapies for underserved patient populations. For access to our research, competitive benchmarking, and best practice frameworks, head over to frost.com or email hello@frost.com. We'll be back soon with more to help you stay clear-headed and growth-ready.